Hi everyone, how are you doing? This is your Anisatagopam. Today's our topic is polymerase chain reaction. Please stay tuned with us and subscribe my channel for further updates. So let's get into the topic. Hi everyone, how are you? So this is your Anisatagopam. So please subscribe my channel and stay tuned with me for further updates. So today's topic is about polymerase chain reaction. So let's start with knowing about polymerase chain reaction. It is known as PCR. It is a widely used laboratory technique. It is used to amplify the detected DNA. So it's not like we'll get the DNA in a large samples, right? So PCR is basically a technique that uses small amount of DNA to multiply into so many strands. Even if we get only one single strand of the dsDNA, so we basically know, right, the DNA is this kind of structure. So even if we get only one certain point, then also we can amplify it and make into a double stranded structure using PCR technique. Okay, it's not like in the crime scenes we always get a large amount of DNA, right? So PCR actually plays a very crucial role at that point. By getting only one strand, we can make into several strands. Okay, let's continue more about PCR. I hope you find it interesting. Yeah. So what is the technique? How we do the PCR? So after, like for example, we got a small strand of DNA. Let's imagine this is the strand. So first what we do is to split the strands. Split the hydrogen bonds in between the strands. I'll show you a detailed thing. Give me a second. So first this is the structure. Just imagine I got this piece, this certain piece. First what I will do is I'll use the heat. At what temperature? 94 to 98 degrees. Okay. So what happens? As a result, it will break this bond. Okay. This bond gets broken, and what happens? Two double strands, two different strands of DNA would be formed before me. Okay. So next we will do is, like, this is known as the fork structure. Okay, replication fork, also known as the, just think it as a replication fork. In the same way as a replication fork, it will also occur for the replication or making into further strands. Okay, so next, annealing. Annealing is the short specific primers. In here, the short specific primers are added complementary to the single-stranded DNA sequence. SS means single-stranded DNA sequences. So now we'll have two different strands, right? Like this. So what we do is we'll put the primers will be added here. And the temperature depends on the primer sequence and GC content. Okay. Next, extension. After adding on extension, Taq polymerase, a thermostable DNA polymerase, synthesizes the new DNA strands. The Taq polymer gets attached here. It starts synthesizing by adding the nucleotides or DNTPs. Complementary, they'll make complementary ATGC, 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 and the strands will be formed. And this will be done in an exponential amplification state, like so many copies of the certain DNA will be formed. Each cycle doubles. Like if in the first cycle I had two strands, right? It became into four, and then that converts into 4 into 2, not 4 into 2, 4 square, that is 16 actually. So then 16 square, and so on, so on, so on. That's how it happens. Okay, I hope you understood that, right? Now let's go ahead and understand this a little bit deeper. So this is how it occurs. So first, at 95 degrees, the denaturation, then the annealing occurs. And the Taq polymer I told you earlier arranges the DNA and it makes the two strands. Can you see that here? There it occurs at 72 degrees Celsius, and in such a way two different strands are formed. Okay, at 95 denaturation, 55 annealing, and 72 extension and formation of different strands. Okay, I'll give you better detailed description. Can you see here? So the strands are separated. In here, can you see the strands 3' and 5'? The strands are separated. How? Denaturation. By giving the heat, the hydrogen bonds are broken and forms two different strands. And later on, this DNTPs or primers are added, and later on what happens is the Taq polymerase keeps on adding the new DNTPs, required DNTPs, and they continue the process. Okay, they go on for extension. This annealing, adding of the primers occur at 68 degrees Celsius, 65 to 68. Then elongation, that is extension or elongation from Taq polymer, that occurs at 72 degrees Celsius. Okay, now let's go further. Once again we'll see it in a detailed structure. So now the denaturation occurred. Now we'll add the primers and the free nucleotides or DNTPs and the polymerase. Now can you see the extension, how it occurs, how the Taq polymerase are making the strands? Yes, at 72. I hope you understood now. This can, and now the types. So anchored PCR, a small sequence of nucleotides can be attached or tagged to the target DNA is called DNA is anchored, or else the DNA is anchored. It's a technique in PCR in which only two ends of the DNA sequence can be amplified. In such cases, anchored PCR may be utilized only one primer instead of two. It's like amplification only of the two ends, not the entire sequence at the ends. Here, one end may be known and another may be unknown. Okay? Okay, this is the technique, how it occurs. You can see here clearly how it occurs. First, the DNA synthesis, adding of poly G, poly C attached to a primer is added and DNA is synthesized. Only one end is synthesized. Okay? And formation of two different strands. After that, the both become adding of primers, and therefore in subsequent cycles, both original primers and anchored primers will be utilized for further application of the target. I hope you understand the class, and please subscribe my channel. Stay tuned with me for further updates. Thank you.